Today's screencast is about cellular respiration. We all know that food is used to give living things energy. We're going to go into the specifics of how that's done. The chemical bonds in food have to be broken, and then they're rearranged so that energy can be formed. Energy that's stored in food is expressed in units of calories. You've seen this on a food label, I'm sure. A calorie, capital C, is the amount of energy needed to raise the temperature of 1 gram of water by 1 degree Celsius. So the calorie here is technically a kilocalorie, what you see on a food label. Cells use all kinds of molecules for food. We've gone into these before, the fats, proteins, and carbohydrates. The energy stored in these molecules varies because their chemical structures are different. Cells break down food molecules gradually and use that energy to produce our magical mystery energy molecule, ATP, for cellular functions and processes. So the process by which ATP is formed is called cellular respiration. Here's the overall chemical equation. Make sure you know that. And if you take a look at it in words, you can see that it's pretty much the reverse of photosynthesis. The cell has to release chemical energy in food molecules gradually. Otherwise, most of the energy would be lost in the form of heat and light. And that's what we see anyways. This isn't a perfectly efficient process. You see the energy being lost in the form of heat and light. There are 3 parts to cellular respiration. The 3 parts are glycolysis, the Krebs cycle, and the electron transport chain. And you can see those over here as we go from top to bottom in this diagram. We'll be looking at this diagram quite a bit. So the first stage is glycolysis. If you look at that word itself, we break it down. Glyco refers to glucose, and lysis means to split, to break apart. And that's literally what's happening here, is glucose is being split into 2 halves. Glycolysis only produces a small amount of energy. Most of glucose's energy remains locked in the product of glycolysis, pyruvic acid, which we'll get into in just a second. So as I mentioned just a couple of minutes ago, glucose is broken down, basically broken in half into 2 molecules of what we call pyruvic acid. And then pyruvic acid moves on to the next stage of cellular respiration, which is the Krebs cycle. In this process, a little bit of ATP and a different electron carrier, NADH, not NADPH. NADPH was the electron carrier in photosynthesis. If you want to think about that, the P would be standing for photosynthesis. It's not NADPH here, it's NADH. Still an electron carrier, but a different one. So the cell deposits 2 ATP molecules to get glycolysis going. We need a little bit of input to get this process going, and then we get an output of ATP, 4 ATP specifically. So the net gain of 2 ATP molecules is what we get out of glycolysis. And again, as I mentioned before, we have NADH, which is carrying high energy electrons, and those go straight to the electron transport chain, and they are used to produce more ATP. So we end up at the end of glycolysis, 2 NADH molecules and a net of 2 ATP. This process happens very fast, and it's an advantage when the energy demands of the cell suddenly increase. Another advantage is glycolysis doesn't require oxygen. It can supply energy to cells when oxygen is unavailable. Now if we move on to the Krebs cycle, a little more energy is generated from pyruvic acid. The electron transport chain produces the bulk of the energy by using oxygen. Oxygen is a powerful electron acceptor, so that's why we need oxygen in order to breathe. The pathways of cellular respiration that require oxygen, are called aerobic. Krebs cycle and electron transport are both aerobic processes, and they take place in the mitochondria. Glycolysis, on the other hand, as we just mentioned, doesn't require oxygen, and it takes place out in the cytoplasm. The word we use to describe glycolysis is anaerobic. It still is part of cellular respiration. It has to happen in order for cellular respiration to get going, and as I just mentioned, it takes place in the cytoplasm of the cell. I made a quick reference to this when I showed you the overall reaction of cellular respiration, but just a little bit more about this here. Photosynthesis and cellular respiration are opposite processes. The energy flows in opposite directions. Photosynthesis deposits energy, creates that availability of energy. Cellular respiration withdraws energy. And the reactants of cellular respiration are the products of photosynthesis and vice versa. The release of energy by cellular respiration takes place in plants, animals, fungi, protists, and most bacteria. Energy capture by photosynthesis occurs only in plants, algae, and some bacteria. So almost every type of living thing does cellular respiration. In fact, I'd challenge you to find a living thing that doesn't. Photosynthesis is a little bit more specialized. So let's take a look in a little bit greater detail at the Krebs cycle. This is also known as the citric acid cycle. Remember that the pyruvic acid that was produced in glycolysis moves on to the Krebs cycle, and it's broken down into carbon dioxide. Here's where carbon dioxide is produced. So if you think back to photosynthesis, the Calvin cycle uses carbon dioxide. In cellular respiration, the Krebs cycle produces carbon dioxide. Again, you don't have to memorize all of these different parts of the citric acid cycle, the Krebs cycle. I'm just going to kind of point out some different overall things. So again, pyruvic acid from glycolysis goes into the matrix, which is the innermost compartment of the mitochondrion. And we're forming more NADH here. And we get our first molecule of carbon dioxide. And there's this intermediary molecule called acetyl-CoA that's formed as we're getting this process started. Now if we take a look at what's going on here, we're moving around the Krebs cycle in this direction. Just some highlights here. So, here we see where our carbon dioxide is being produced. Here we see, and carbon dioxide here as well. And then, so we see that here also, we're circling around. And then, I don't know why all of these aren't colored in, but we have electron carriers here that are getting sent off to the electron transport chain. And then we also have ATP being produced here. Now, the thing to keep in mind is we have two pyruvic acids. So the cycle turns twice. We have two pyruvic acids that run through this Krebs cycle. And that's what I have indicated here on this slide. 2 molecules of pyruvic acid enter the Krebs cycle. Each molecule of glucose will turn the Krebs cycle twice. So just in terms of totals, for each glucose molecule, 6 carbon dioxide molecules, 1, 2, 3, and then turning twice, 4, 5, 6. 2 ATP molecules. The ATP is right here. So that's going to be 2 of those. 8 NADH. So there's some here, 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 and here. So that's 8 of those. And then 2 FADH2 molecules are produced. Now, all of these here, the electron carriers, will go to electron transport. And that's what you see indicated here. 2 electron transport chain, 2 electron transport chain, here, 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 and here. And that's what we'll talk about next. So here we have the NADH and the FADH2. They've come from glycolysis and from the Krebs cycle. They dump their high energy electrons into the electron transport chain. At the end of the electron transport chain, these electrons combine with hydrogen ions and oxygen to form water. This is why we exhale water vapor. Energy generated by the electron transport chain is used to move hydrogen ions across the inner mitochondrial membrane and into the intermembrane space. If you remember, what was going on here, or what you see going on here, is very similar to what was happening in the chloroplast in the light-dependent reactions. Because look what's happening then at the end. Hydrogen ions pass back across the membrane through the ATP synthase. Here's our ATP synthase enzyme, right here. Here's ATP being formed. I colored it too much, now you can't see it, sorry. There. With each rotation, the ATP synthase attaches a phosphate to ADP to produce ATP. And again, you don't have to understand the how or the why that happens, but just know that that's how ATP is produced, and ATP is our magical mystery energy molecule. So let's total it up. The complete breakdown of 1 molecule of glucose in the presence of oxygen, so aerobic cellular respiration, gives us 36 ATP molecules. That's about 36% of the total energy of glucose. The remaining 64% is lost as heat. So we've used glucose as our example, but anything can be broken down and rearranged into simple sugars like glucose. Lipids, proteins can be broken down, complex carbohydrates can be broken down. The cell can generate ATP from just about any food source. We just kind of kept it simple by starting with glucose. Now, if there's not oxygen available, we can still produce ATP. It's a process called fermentation. It occurs in the cytoplasm of cells. So again, this is anaerobic conditions. Basically, fermentation allows glycolysis to continue. And that ATP, that small amount of ATP, that net gain of 2 ATP, which is not nearly as much as 36 ATP if there's oxygen, that net gain of 2 ATP can be continued. There are 2 main types of fermentation. One is called alcoholic fermentation. Yeast and a few other microorganisms use this process, and it produces alcohol, ethanol, ethyl alcohol, and carbon dioxide. This process is the basis for producing alcoholic beverages, and it causes bread dough to rise. Now, before you think that there's alcohol in bread, the bread is baked in the oven. The heat evaporates the alcohol off. So there's not alcohol in bread. Here's the chemical equation for alcoholic fermentation. So here we have our alcohol and our carbon dioxide. And again, this process allows glycolysis to just continue to cycle. Lactic acid fermentation happens in humans and some bacteria. This converts pyruvic acid to lactic acid. Here's the chemical equation. And again, we're allowing glycolysis to continue. So it makes sort of a cycle. Lactic acid fermentation is the basis for production of some sorts of fermented dairy products like yogurt and cheese, sour cream. Those are produced through the process of lactic acid fermentation in bacteria, lactobacillus. So cells normally, again, living things are lazy. They have small amounts of ATP, enough for a few seconds of intense activity. Lactic acid fermentation then can supply enough ATP to last for about 90 seconds. However, extra oxygen is required to get rid of the lactic acid produced. So following intense exercise, a person will breathe heavily to pay back what we call the oxygen debt and clear lactic acid from the body. It's thought that lactic acid is a cause of muscle soreness. As we're looking more at this, the farther along we go in research, it appears that it's a combination of both lactic acid and just small micro tears in the muscles. For intense exercise lasting longer than 90 seconds, cellular respiration is required to continue that production of ATP. Cellular respiration releases energy more slowly than fermentation. The body stores energy in the form of the carbohydrate glycogen. Glycogen stores are enough to last for 15 to 20 minutes. After that, then the body begins to break down other stored molecules like fats for energy. So if a person wants to lose weight, they need to, and break into that fat supply, they need to exercise for longer than 15 or 20 minutes. Animals that hibernate, like the bear, rely on that stored fat for energy when they sleep through the winter.